Hello students, today we will see chemiosmotic hypothesis from the photosynthesis chapter. Okay, let's start. In this chemiosmotic hypothesis, first we will start with the chloroplast. So, this is the chloroplast structure. Clear? So, now one more membrane. Then inside that we have thylakoids, stack of coins, correct? And then they comprise to form grana and then connecting is stroma lamella. So this particular thylakoid is zoomed, okay, is zoomed and drawn over here. This is the zoomed over here, okay, and then two membranes we have. So here only we are going to see the chemiosmotic hypothesis. So be clear with this. This particular thylakoid, on the particular thylakoid membrane only the chemiosmotic hypothesis takes place. So I am taking only one thylakoid, okay, be clear with that. Let's start. This is one thylakoid, this is lumen. This is the thylakoid membrane, okay, this is thylakoid, one thylakoid, and this is the thylakoid membrane, okay. Here we have photosystem 2, clear, and here we have photosystem 1. Between that we have cytochrome complex, clear, this is cytochrome complex, okay. When light falls on this and this, the electrons getting excited, so light comes here and the electron reaches this and again comes over here, and okay, for this step will stop. Okay, we have seen that PS2 over here, cytochrome complex PS1. Here we have one more thing, it is NADP reductase complex, okay, and here we have water splitting complex. This is water splitting complex. I will write here. Water splitting complex means the photolysis of water takes place over here, okay. So now here electron comes down, then it reaches over here and forms one NADPH2, correct. I got this NADP plus and one NADPH2 is formed, okay. Now where this hydrogen comes from, that we will see now. That's what chemiosmotic hypothesis. So in cyclic or non-cyclic photophosphorylation, electron excited from PS2 and PS1, the carriers cytochrome complex carry the electrons to PS1, and this electron again jumps to NADP reductase and NADPH2 forms. This is a process of cyclic non-cyclic photophosphorylation, okay. This hydrogen is gaining here, reduced, no. Where from this hydrogen comes? Okay, two process we are going to see now. What are those two processes? See here, this cytochrome complex, when carrying this electron, okay, carry a pair of proton. While carrying one electron, a pair of proton comes inside, okay, pair of proton comes inside, clear. So from stroma comes inside, this is stroma. So this white board is stroma, this is a particular thylakoid, okay. Inside this thylakoid we have lumen. So in this lumen, one process is in the cyclic or non-cyclic photophosphorylation, the carrying of electrons, the carriers of electron, carry the hydrogen, while carrying the electron, carry one pair of hydrogen from the stroma to the lumen. Then the other process, here we told that water splitting complex. Here H2O, okay, H2O is broken into H plus O2 and plus 2 electrons, 2H plus half O2. Okay, this is our compounds products when one H2O is broken down inside inner membrane of the thylakoid. Okay, so this H plus accumulates over here itself, and this goes to the atmosphere. Then goes to fulfill the deficiency of the photosystems. So the other process, one process is in the process of cyclic or non-cyclic photosynthesis, and the second process is the water photolysis of water gives hydrogen in this lumen. So hydrogen, this is increased, more concentrated inside. Okay, and here we have one more structure, that is F0 and F1 complex. This is together known as F0 F1 complex. Okay, here, so I told you, know, where from, I asked you a question, where from this hydrogen comes from. That hydrogen comes from this complex. Okay, so this thylakoid membrane is impermeable to H plus ions. So this thylakoid membrane is impermeable. Okay, permeable to H plus, this one. Okay, so this carriers only carry the hydrogen from the lumen to the outside, to the stroma. Okay, while they carry hydrogen from inside to outside, they form an ATP in the result, ADP plus inorganic phosphate. Clear? So when take proton from outside to inside, okay, so they form, the energy lost by this electron is used for the synthesis of ATP. Clear? So this hydrogen, when come to stroma, it goes to NADP reductase for the formation of NADPH2. Okay, clear? This is chemiosmotic hypothesis. Simple concept, only two processes important. What is the two process? One thing, in the cyclic or non-cyclic photophosphorylation, the H plus ions comes from the stroma to the lumen. So one process here it takes place, and the other process here it takes place. The water splitting complex is there. They split the water, and these are the products formed. So electrons goes to the fulfillment of the deficiency of photosystem, and the oxygen goes to the atmosphere, and hydrogen accumulates over here. So hydrogen concentration is more in lumen. So here hydrogen in stroma decreased, correct? So to fulfill that, the hydrogen comes through the F0 F1 complex, and while carrying the hydrogen to the stroma, ATP is formed. Okay, this hydrogen is used in the formation of NADPH2. That's it, chemiosmotic hypothesis. Okay, children, clear? If you have any doubt, comment in the comment section, and subscribe our channel for more videos, children.